All right, everyone, let's get ready to get started. Can I get someone to confirm that you can hear my audio? All right, thank you very much. So hello, everyone. My name is David Ruck. I'm here at the University of Maryland in Baltimore, Maryland, and welcome to Cytometry in R. And today is a very exciting class. We are first being exposed to the different Tidyverse packages, which we're going to be using consistently throughout the course. And collectively, the Tidyverse packages have enabled R to be a much more beginner-friendly language, a much more usable language than what it was 15 years ago when we only had base R. So, for a little bit of background about why this is such an exciting thing to encounter, let's think of our daily workflows as flow cytometrists. So after putting all that effort, building your panel, actually acquiring the data, we on our different instruments, we need to actually go and take them back to the lab where we start working on our gating schemes. So we set up our gates, we set it for every cell population that we're interested in, and at a certain point in our workflow, we need to export out the data from those gates that we drew, and then take that data and start doing downstream statistical analysis with it. Now, if you're anything like me, when I first started my PhD program, a substantial amount of my day was spent renaming column names of the exported out FlowGeol data, just so that it would fit nicely within a single Microsoft Excel sheet column. Additionally, I would set up formulas which basically combined the proportion of the positive quadrants so we know how many cells actually made interfering gamma, not just what was interfering gamma, TNF alpha, double positive. And then finally, after those additional hours went by, I would then spend additional hours copying and pasting over these nicely organized columns over into GraphPad Prism so I could then run individual statistical analysis for each comparison. So this would be in the ideal scenario. However, oftentimes the data was far less organized, and I would end up with data that had columns with like intermediate rows appearing as empty spaces. So a good portion of time would then be spent copying and pasting over individual cells within the worksheet until everything was in a single column, all the while trying to remember what are these particular color codes or highlights or the bold font that I had drawn in for this particular Excel sheet a couple months ago, what is actually going on at that case. So today we're going to explore how to make our data sets tidy and what exactly does this mean. I linked the paper by Hadley Wickham that goes into the actual ethos as far as why it's advantageous to treat everything as far as in terms of rows and columns. But the very simplest, if we're trying to understand what these Tidyverse R packages are working for, what they're revolving around, it's the concept that if you turn all your data into essentially table-like objects, so everything being a square or everything being a rectangle, at that point, if you think of it in terms of just rows and columns, you need substantially fewer number of tools, i.e. the functions we use in R, to be able to reshape this data and extract out the useful information that you actually need for your statistical analysis. Additionally, there's an advantage of thinking of everything in terms of rows and columns because this is much closer to how our computers actually operate and interact with the data. So what data management tasks that would have taken us hours by hand to be able to adjust everything over would take the computers just mere seconds or just a couple minutes to be able to do the same equivalent task. So the dataset that we're going to be working with is one that I generated. It's based off of manually gated spectral flow cytometry dataset for a HIV-exposed uninfected infant cohort. So it has different conditions as far as the different treatments that individual tubes for the experiment were treated with. It has different time points ranging from cord blood through 4 and 9 month blood draws from peripheral blood. It's very much, if you look at this initial data set, it's messy, and I left it intentionally messy. It's not been taken all the way to being a ready-to-go for statistical analysis kind of data set. However, if you wanted to work with a different data set, you could just as easily swap in a matrix or a data frame that we learned how to extract from a FCS file last week, or if you have your own data, either exported through whichever commercial software you're using, as far as the output, you could just as easily switch that in for today's lesson and just do it with your own data set. You just need to make sure you set up the file path to point at your particular file, not the actual data set that we provided for the course. As always, there's a little bit of homework, housekeeping. As we do every week, I've updated the GitHub to contain the new materials for this week. So please go ahead and sync your forked version of the Cytometry in R folder, and then bring everything local into Positron doing the pull request. And if you're keeping the organization scheme of version control that we've been doing throughout the course, create a new week 4 project folder, copy the contents from the Cytometry in R folder over to your particular folder so that no edits that you do affect everything that you're working with. And then once you have everything set up, you've set up your subfolders for images, data, etc., however you're keeping your everything organized. Go ahead and make that initial commit so everything is kept within the version control software, and go ahead and push those changes to GitHub so that your data set is available and your code is available, like worst case something should happen to your computer. Additionally, for those of you who have been submitting the take-home problems, we encountered a bug with being able to bring in the data for the subsequent week. I posted a link to the walkthrough to how to solve the issue as far as being able to bring in the data. And then starting next week, we'll have a slightly different protocol as far as submitting the take-home problems, just because you'll need to ultimately be, instead of pulling a pull request from your main fork in Cytometry in R to our homework branch, you're going to have to create your own branch, and then it's going to be a branch-to-branch transfer. But the details of that are explained within the walkthrough. All right, and with that, let us begin. So the file we're working with today is called, of our data set, is called dataset.csv. So obviously it's a CSV file. You can do the same kind of procedures with Excel sheets, but you just need to change the function that's actually doing the handling. And currently, if you're keeping the organization scheme from throughout the course, our file path to locate that specific file is going to look something like this. And you'll notice this file path has changed slightly. So instead of just pointing at a particular folder, I'm actually specifying the file itself within the file path. So it's kind of similar to what we would have gotten as an output if we had followed through with the subsequent step of doing the list.files. So the reason I encourage everyone to use the file path function when they are generating their file paths is it's much better as far as being able to keep everything within your code base, both reproducible and replicable. So if you have to transfer your code over to a friend or a supervisor, doing all your file paths dynamically with the file.path function will mean that if they're using a different computer operating system, you won't have any hard-coded slash, either forward slash or backward slash, that are going to interfere with their ability to run your code. And you're not going to have them search down all the backward slashes or forward slashes and switch it out at that point. So as much as possible, try to use the file path when you're trying to locate particular files. Now, as I was mentioning just briefly, I provided the actual file name that we were interested in. So both the file name itself, in this case dataset, but also the file type as the last argument within that file path function. So it was quotation marks dataset.csv. So this allows us to basically bypass that step that we did as far as finding list.files and then matching a pattern and then identifying that particular index that we wanted to bring in. The tricky part is if you accidentally mistype the file name, as you go through the subsequent steps, you're going to encounter an issue where it's going to say no such file found. And it's not because your file is not there, and it's not because your working directory is in the wrong location. It's just that you misspelled the file path, and that's causing the error that you're encountering. So, since our dataset is located within a CSV file, we need to use a function that is capable of working with CSV files. So the one we'll be using today is the read.csv function. Now the read.csv function is included within the utils package, which is part of the base R software installation. So everyone has it. You don't have to call library to be able to load it in. Now, read.csv may not be the function you want to keep around long term if you're reading in much larger CSV files. There's different packages in R, including the reader package and the data.table package, which do a lot better and faster job at reading in CSV files. Similarly is the case with the arrow package. So as we get further on in the course, you might want to explore those alternatives to be able to bring in the data slightly faster. But for today, I know everyone has it, and I know it works well, and it also highlights a couple other things we should keep an eye on as beginners first learning how to use R. So we'll be using it for today as our example. So the way this read.csv function works is kind of similar to everything we've been encountering so far. we need to provide it a file path to whatever file we want to bring into R. In this case, we already designated that as an outside argument, outside variable, and outside object. So in this case, the file path we defined on the last screen. And then read.csv has a second argument, the check.names argument, which in this case, we're leaving it set to false. I'll show you what happens when we set this to true in just a couple minutes. So at this point, R goes and searches your file path. It uses the read.csv function to read in all the underlying data, and then we're using the assignment arrow to assign it to a new variable or a new object within our working environment, which we're calling data. Now, if we use the column names function, kind of similar to what we did last week, we can just retrieve from that object all the column names that are present for the particular CSV data set that we just loaded in. So in this case, we can see the column names are printed as a vector. And what we can see is up on the upper portion, it looks to be mostly like metadata columns. We can see there's the time point column, condition, date, sex, a couple others that might be related to the specimen itself. Then we start having more traditional like flow cytometry names for column names, singlets, CD45, T cells, etc. But then we also encounter what we normally expect for our particular flow cytometry gates. There's V delta 2 positive, V delta 2 negative, CD8 positive, CD8 negative. And there's a couple that also may remind us they might actually be actual counts instead of like the proportion or the frequency. In case we look at, there's lymphocyte count and a CD45 underscore count column. So this is what happens when we read in the data with the check.names argument set to false. But what happens if we go ahead and switch that over and set it to true? So let's go ahead and do that. And when we check, we can see that in this version, with this extra argument set to true, we still get the same kind of metadata column names. But as we get down to those more FCS file kind of column names, we see something weird has happened. We are no longer seeing the plus and minuses, and we're no longer seeing anything with a space. At this point, they have been replaced by dots, as well as .1 or .2, depending on what's going on here. So what's happening is, remember, R does not like spaces, and R does not like having special characters within the name. That's part of the reason why in the last week, when we looked at those keywords within the FCS file, those that included a $sign needed to be surrounded by quotation marks. So what's basically happening in this case here is the check.names argument within read.csv is basically verifying are the column names according to how R prefers to have its syntax. If it encounters one that doesn't match the syntax, it has special characters or it has a space, it will just autocorrect it and place in a R appropriate character. Now, as you can see, this is not necessarily the appropriate choice in this case. R did something broad in general that applied across the board. At this point, we don't actually know is this CD4 dot or the CD4 .1, the CD4 positive or the CD1 negative gate. So this is a situation where just because R can do something by default doesn't mean it's necessarily what you want to do. So in this case, I recommend if you do have a column name with a special character and you want to rename it in an R appropriate fashion, use a different function. Don't use this broad general one when you're actually going about and doing that. And we'll learn more about how to do this a little bit later today with the rename function from the Tidyverse. All right, so at this point, it's also good once you have read in your first data set just to get a good general idea of what is actually happening with that data. So this is where that head function we encountered last week can come into play. So I just want to see the first 3 rows of our particular data. And we can see bid column looks to be something kind of related to the specimen IDs. We can see time point corresponding to the cord blood, the 4-month and 9-month blood draw, conditions, date, a few extra things. We can see for our data, there's things that look like actual cell counts, but there's also the actual proportion of probably the parent gate. So this is an example as far as what I mean. Some of this is not quite clean. It's still a little bit messy. It's definitely something that if you're trying to work with, you want to be able to polish it up, tidy it up a little bit so that you're able to glean much quicker the actual information that you care about. So when we're working within positron, just like there's always multiple ways of doing something in R, if you have already created that data object, that data variable within your environment, we can do the same process as far as what we did last week. And if it's a matrix or if it's a data frame, if you click on the little grid, it'll pop up and open in your editor window. And at that point, you can do the same kind of exploration, but just look at everything across the board. You'll see in this case, it's 196 rows total, and there's like 31 different columns. You could also have achieved the same opening up of the data in the editor window using the view command from the console, and that would have looked something like this particular one. So, just like we covered last week, functions in R are the analogy of having tools in your toolbox. And just because you have a tool doesn't mean you can use the same kind of types of things to be able to do the same kind of job that you need. So just like a hammer needs a nail, a screwdriver needs a screw, a paintbrush needs a paintbrush, in R, your different functions, which act like tools, need certain types of objects on which they're able to carry out their work. Some of them may require a vector, some of them may require a matrix, some of them may require a data frame, some may need character values. So whenever you're loading something into R for the first time, just double check to see what kind of type of object are you working with, which we can do with the functions we learned last week, the class function and the str, which stands for structure. So if we quickly check data, we can see that it's one of those data frame functions, not one of those data frame objects, which as we saw last week is kind of like a matrix, but is able to handle a little bit more difference as far as character columns, logical columns, etc., etc. And then when we look at str, we can get a much more detailed view back. We can see the actual column names are after the little dollar sign. We can kind of infer the values, the types of values that are present for each of these columns because we see character and integer values being returned. And then we can see some of the underlying data being displayed, but you'll notice it's not quite uniform, it's not quite aligned, it's a little spread out across the board. So, when we're working with a data frame, we can also be able to check to see what contents are in it using the glimpse function. However, when we try to run glimpse just as is right now, we'll get back this error saying it's not able to find the function glimpse. Now, this is a situation that we have experienced in the past, but not exactly in the same form. We know that from the past when we tried to install packages and or install something from Bioconductor, but we had not yet loaded the Bioc Manager package, we're getting a similar type of error. So in this case, we can surmise that the glimpse function is not in base R. It's likely within one of the other packages. So it's like having a toolbox, and we're trying to find a tool without having yet opened the toolbox. So we're going to need to open the correct toolbox in order to find the glimpse function. However, we don't know which of the many toolboxes we've installed into our local computer at this point actually contains the glimpse function. So what we can do is we can basically tell R, check all installed packages and check for anyone that has a glimpse function in it. And the way we do that is very similar to what we've encountered in the past. Namely, we're just going to add an extra question mark in front, in which case we have double question marks glimpse, which is just basically going to look within all our installed packages until it finds any package that has glimpse as a function name inside, and it's going to return us a list of results as far as each of the packages go. And then it's up to us when we're looking at the list of returns just to decide, okay, is glimpse from the dplyr package or is it from this other package that may not be related to what we're trying to do? So this is kind of like how you would work out where a function is from if you don't actually know which package it's currently within. So from that list, we can ultimately surmise it's likely coming from the dplyr package, which is the main Tidyverse package that we'll be going through throughout the course and the main one we'll be covering today. So let's go ahead and actually attach dplyr to a local environment so that we can actually be able to use it. And as a reminder, we're doing this through the library call. And once we have loaded library, if we use glimpse data again, we see actually we get the values back. So as far as comparison to str, it's very similar. We do have the difference of the rows and the columns are listed individually at the top, but then you can see the spacing has changed a little bit. It's a little bit more uniform across the board. Now, if we had just wanted to retrieve the dimensions of the particular data frame, knowing how many rows and how many columns, well, we could do that visually and just by checking it, either through the positron window or through glimpse. We could also do it using the dim function, which stands for dimensions, which is going to return to us basically this number of rows and number of columns based on the original base R notation. And in base R, as I remind you from last week, the number of rows is always before the comma, and then the number of columns are always after the comma. So if we run the dimension function as is currently, we see it returns number of rows being 196 and number of columns being 31. So as we saw, we already kind of glanced and we saw some of the types of values that are located within each of the columns, whether they're numeric, whether they're character values, or whether they are logical, meaning they are true or false. However, when we look at the data again, we see there's a little bit of variation. So some of these numeric values are appearing as integer as far as the type, while others are appearing as double. So within numerics, there's these two subclasses of numeric type values, basically corresponding to, are they whole numbers or do they contain a decimal point? So in the case of containing a decimal point, they're being stored as double as the type of value, versus the integers are the whole numbers in this case. So that's just another one to keep an eye on as we go forward. Now, just like everything in R, there's always a way of checking to see if something is a particular type of object within the console window without having to rely on things visually. So in this case, these are three very similar related functions: is.character, is.numeric, and is.logical. So if we were wanting to check what kind of values are present within our bid column, just like we learned last week, we can actually select that bid column by using the little $sign accessor, which is basically just going to look at the contents of bid. And then when we pass them to the is.character function, it's going to check those values that are present and tell us, are these values that are being seen character values, true or false at that point? So in this case, it'll come back as true. However, if we had done the same exact thing, but used the is.numeric function, checking the bid column, which corresponds to the specimen IDs, it's going to come back as false because there are character values present. And a similar story happens like if we were checking the T-cell count. So using the little $sign accessor within data, we're looking at the T-cell count column. But when we're checking to see if the values within the count column are character values, we can see that they're returning as false, meaning they're likely numerics of some form. And just like numeric has those two subtypes, there's the variations of, is it an integer value that's present within the type, or is it a double as far as the type of value contained within the columns? So checking counts, you can see that it's coming back as true as numeric, which is the broader overview. It's also coming back as true for integer, but when we're checking to see if it has a decimal point and the is double, we see it's coming back as false at this point. So, as always, just a reminder, if you're working with the column names that contain special characters like $, ampersand, exclamation points, or spaces, if you're actually trying to access those column names, you need to surround the column name with a click mark. So if we try to run the CD8 negative column as currently is, we're doing data, and then we're using the $sign accessor to get at the CD8 negative, but we get back this error when we try to run is numeric. And if you look at this and try to read what's going on, it looks like R is currently trying to interpret this as a subtraction. Like you're trying to subtract that column from something else. So in this case, this is a situation that that special character gets recognized as being used in an alternate way at this point. So if we actually wanted to check, is the CD8 negative, are the values within that CD8 negative column numeric, we just need to surround it with the tick marks or the quotation marks just to be able to tell R, hey, this is a column name, don't treat it like a special character, which would be a code. All right, now that we have read in our data, and we have a good overview of what the different columns are, and what the general types of values contained within those columns, we can actually start working on learning how to use the different dplyr functions that we need to keep our data tidy. So once we have called library, we should have access to the select function. And basically, the select function is going to select a column within your particular data frame and isolate it out as its own separate object. So what you'll see often is select is going to have within its parentheses two arguments. The first argument corresponds to the data frame or matrix that you're trying to select columns from. And then that second argument is basically going to be the column name that you want to isolate, that you want to separate from the much larger object. So you can see when I'm using the date columns, for now it's just surrounding it in quotation marks. It's actually selecting out the date column and leaving all the other columns that we weren't interested in still in the original data frame. In this case, we've subsetted it to a new object, which we're calling the date column data frame. However, if you're actually reading code that other people have placed out and made available on GitHub or in a paper, you'll rarely see select used in a line of code in this exact same format. And the reason for that is within Tidyverse, there's what's known as pipe operators. So oftentimes you'll see it actually being displayed in this manner, where you have data and then you have this pipe greater than sign, select, and instead of having two arguments in the middle, you just have a single argument. Now, if you're anything like me, your first reaction to this was, what in the world is this thing, this pipe with a greater than sign next to it? So, within Tidyverse packages especially, they use extensively what is known as pipe operators. So if you have had previous R classes, you'll be more familiar with the old Magritter style package pipe, which was the percentage greater than percentage sign. Or if you're just starting off, you may have encountered the base R version, which was instituted after R version 4.1, which is just the little pipe with a greater than sign. So if you're actually like looking at it and seeing lines of code, you'll see these two different variants that basically are accomplishing the same task. And we'll talk a little bit about the difference between these two in just a little bit. So how do we actually try to interpret this pipe thing when it's present within our line of code? So let's go ahead and break this down slowly. And let's start off first just to the right of our assignment arrow, just as we start working with our data object. So when we're working with data, we know from having explored around that this is a data frame. It contains column names and contains different columns that contain different types of values. Now, when we're passing it over to a pipe operator, we're basically letting the pipe take all the contents that is immediately to the left in this type of object and make it available to whatever the function is going to be right afterwards. So in this case, if we're using select and we're passing, what's going to be happening in this line of code is within data, it's everything that we're actually interested in. And through the pipe operator, we're making it available to the select function. So in this case, select has the data already accessible, already available. So we don't actually need to specify that first argument which corresponded to our data frame or a matrix. It becomes available automatically. So the only thing we need to provide to select at that point is just the column name that we actually want to select, that we actually want to isolate from the much larger data overall. So if we're completing this, we'd see in this case, if we're using the quotation marks, select has within the parentheses quotation mark state. And then if we ran that same line of code, just as we saw before, we would isolate out the date column from the overall one. And then if we're doing an assignment arrow, it would be passed off to a new object, a new variable that we're creating within our environment. Now, some of you may be asking, okay, it does the exact same thing as that first way, but what particular advantage may be using these pipes actually have? And the particular advantage is pipes can be chained together, linking different functions and outputs of those functions onto the next function that's going to be using them. So, for example, let's say we had just done this one here. At the end of this function step, we have isolated out the data column. We have not assigned it back to a particular object yet at this point. But if we put in another pipe and we're passing it to the next function, in this case the nRow function, which is just going to take our initial column that we have and count the number of rows present, it's just going to go straight back to giving us the actual output that we're interested in and not providing us that intermediate column object that we then have to assign to a new variable. So in terms of the actual advantage, it's saving us having to write this as two separate lines. First, data pipe select and assigning it to a particular object, but then that particular object once again being passed to the number of rows just to get to that same value output that we actually want. So it can be quite useful if you are only interested in a particular thing from coming out of your data frame, just to put multiple of these pipes together until you get to that particular object that you're interested in, and it's shaped in the particular way that you want it shaped, at which point you can save it as a variable in your environment without having to have 15 or 20 intermediate step objects cluttering everything up. And you'll gain a little bit of familiarity with this throughout today as we use pipes a little bit more. Now, for those of you who have had an R course before, you may have seen the old Magritter style pipe, the percentage greater than percentage pipe. And you may be curious as far as like the new base R pipe that was implemented in R version 4.1 and greater, what is actually the difference? So at this point of the course, they're practically interchangeable as far as what we're doing for passing objects onto the next function. There are a few nuances, which I've linked to, that come into play, especially as we get further on in the course, when we start doing more advanced use cases, when we start using like periods within a particular iterator name. So we'll come to this as we encounter them. I will tend to default to using the new base R pipe, the little pipe with greater than sign. However, if you have the old version already hotkeyed within your particular environment, if you're just more familiar with it and want to continue using it, you can perfectly feel free to do so. You'll just see the code in my case more often than not is going to be a pipe than greater than sign, unless we encounter one of those advanced use cases, in which case I will use the greater pipe. Now, if you were tinkering with this on your own, you'd have encountered an odd R behavior, which is going to be the first of many R quirks that we'll encounter throughout the course. Namely, as I was mentioning, we are placing the column name between quotation marks, but if you had forgotten to include your quotation marks, it would have affected anything, and you would have still selected out the column in question. This is very different to the behavior we saw with install.packages, where if we forgot to include the quotation marks, it searched our active environment, saw there was no object, no variable called dplyr, and then would come out and give us an error. So what's happening in this case, where if we forget to include quotation marks on the column name, it's not actually returning. So what's happening in this case is basically can be thought of dplyr, R package, and the Tidyverse packages in general are kind of picking up the slack. Now, the actual description of what's going on is nuanced and it's for another day, and I have included the link for those who are interested in. But basically what's happening is the Tidyverse packages are able to infer, based on what's being passed to the pipe, that you're working with a data frame and that therefore that particular name that you're providing is likely related to a column name of that object that you're working with, not something within the environment. So that's the reason why this particular odd quirk that we encounter here is not returning us an error, but is actually proceeding on. It's because of something that Tidyverse packages are doing on the back end, not because of something changing as far as your general R environment. All right, so at this point we've used select to select just a single column as date. But similar thinking like what we've done previously with the base R syntax, where if we do the little square brackets, comma, then 2 to 5, we select multiple columns. Is this Tidyverse select function actually going to be able to select multiple columns? And the answer is yes. So if within our select function, being passed over the data from the pipe, you start listing multiple column names, in this case bid, which kind of corresponds to the specimen, time point, condition, T cells, as well as CD8 positive, CD4 positive, noting these have special characters, so they need their little tick marks. You can see from the big broad data frame, it gets subsetted down, so it's just those columns that we have specified that we actually want to keep available. Similarly, if we had changed the order of the columns within the column names within select, you'll notice it rearranges the columns based on how we had asked it to do. So in this case, it's putting T cells as the second argument, CD8 and CD4 as the third and fourth columns respectively, as far as what we're getting. Now, this could be useful, but if you have a very large data set with like 199 columns, you don't actually want to have to like rewrite out all 198 column names within your select function just to move that very last actual column name. So what you can do is a different function from the dplyr package, which is the relocate function. And similar to what we saw for select, the data is being passed over from data through the pipe to the relocate function. And that makes it available so that relocate can work with it. In this case, as far as the syntax, we're specifying we want the column named T cells, and basically we want to place it dot after being the second argument, and then we just specify the column name of the column we want to place after in this case. So you can see T cells moved from wherever it was in our original data, and it's now being placed right after bid. And you'll notice I keep adding additional pipe right afterwards, where I'm passing it to the head function, and I'm subsetting down just to the first 5 rows. This is not something you'd be doing with your actual data, but there's only so much space I can show within this particular slide for this particular presentation. So I don't want to have this all displaying all 196 different rows. So I'm just adding this there after all the different lines of code for today, just to keep it manageable and visible within the screen window, not because this is what you would do for your actual analysis. And just like we saw with select, you can also rename multiple columns at once. The only thing you need to do is include a comma between each of the column names. So in this case, if we didn't want to just relocate T cells after bit, but we also wanted to relocate monocytes, we just add a comma and place that second column name in. And then as you can see, T cells and monocytes have now been moved over to where they actually need to be located at. And we can continue modifying arguments as well. So we don't have to place everything after a particular column. We can modify this to place something before. So if we wanted to place the T cell column before the date column, we just change in the argument, and you can see now T cells is appearing before date. And likewise, if you don't remember where exactly a column name is located or what the column name is, you can specify a column index. So if you wanted the date column to be the very first column in your actual object, you just say place it before, and this basically corresponds to column index 1. And as you can see, date has now been moved to the very first column. As we mentioned before, there's the ability to need to be able to rename, especially if you have special characters or spaces in the column name. This is going to be a situation where if you're trying to do it in base R, it would have been much more complicated of a process. So if you think about in our context of our particular data frame, we have CD8 positive, CD4 positive, as far as column names, that contain special characters. So if you're trying to rename this using base R, the way you would have done it is you would have specified your column names and then designated the index. So CD8 is the third value in, so it's placed between the square brackets, and that's returning us as CD8 positive. And then the way you would have renamed the column name back in base R is then using the assignment arrow, basically overwriting the current contents of column name subset's third index, and just kind of swapping it in for CD8 positive at that point. Now, you don't have to be going through all that extra extent when it comes to the dplyr package as far as its rename function. The way this actually works is kind of similar to what we saw for relocate, where whatever data we're having available is being passed to the pipe through to rename, which is then able to act on it. And then the old column name is placed after the little equal sign. In this case, because CD4 positive has that special character, I'm surrounding it with quotation marks. And then whatever new column name we want to give it is appearing to the left of the equal sign. So in this case, you can see when we rerun column names, we see that CD4 positive has been renamed as CD4 underscore positive. Similarly, as what we saw for both select and relocate, you can rename multiple things at once. You just have to remember to include the commas. So in this case, basically we're saying the rename function, the bid column, I want to rename it as specimen. The time point column, I want to rename this as time point underscore months. The condition column, I want to rename this as stimulation. And the CD4 positive column, I want to rename it as CD4 positive. And then if you run that line of code, you can see all those changes to the column names are automatically carried out in relatively quick fashion. Now, sometimes we may not want to be retrieving just the column like we did with select, but we actually might want to contain the individual values that are within that column, either as a vector or as a list. So this is where the pull function would actually be used. And basically what happens is similar to what you have with select, it grabs that particular column, but instead of keeping all the individual rows, it just pulls them out and places them within a vector. And then you can see we have all these made available at that point in time. Now, oftentimes what you'll end up doing for pull is you're trying to identify how many unique values are present. So if you know for a particular condition, there's going to be 3 conditions. In the case of this data set, control, PPD stimulated, SEB stimulated. If you were trying to figure out is there a 4th condition as far as like PMA or some other antigen, how will you go about checking it? Well, if you ended up using pull, you can isolate out the vector of all these values, some of them that are repeated. But if we pass it on to the next pipe to the unique function, it will only keep the first instance of a particular object that it encounters. So you can see that the experiments within this data set we provided were acquired basically between late July and late August of last year. So this is one of those use cases of pull in combination with the unique function. Now, so far, everything we've been working with has been mostly focused on columns. We've used select, we've used pull, we've used rename, we use relocate, and as we mentioned, tidy data is very much concerned with both columns as well as rows. So what's one of those functions that we'd use if we're like trying to like sort or select things that are based on contents within a particular row? And that's where the filter function is going to come into play. So, as I mentioned, within our condition column for this data set, we have different FCS tubes that were stimulated with different conditions of antigens. So we have a baseline for control. We have PPD to check whether they were TB vaccinated, and then we have SEB just to measure broad polyclonal responses for activation for our T cells. So if we wanted to take that big data frame, which has 197 rows, and subset it down just to look at either the PPD or the SEB that had the rows that contain PPD or SEB within that condition column, how would we go about doing that? Now, if you were working within Excel, you could open up the column, do a little check mark, set it for a particular value, and then that would subset down to your particular view. So in R, we have something similar, namely the filter function from dplyr. And the way you would see the syntax set up is, once again, the data is being made available to the pipe, to the filter function. And then what it's doing is looking within the condition column. It's matching anything that's equal to, which is being designated by these two equal sign. And then we're surrounding the particular value within the quotation marks, meaning PPD. So if we were to run this line of code, we'd see that only things that had PPD within the condition column are being kept in the actual data frame itself. So we're subsetting down the total number of rows that we actually have present. Now, using the double equal sign worked particularly well in this case. However, it can be glitchy, especially in practice with character values. So I don't recommend setting up your filter functions like this. More often than not, you'd be better using off the percentage in percentage operator, which does the exact same thing, but it's able to account for those edge cases that you'll encounter with character values. So if we just rewrote that line using the safer approach of doing it, we'd have the data being passed through the pipe to the filter function. It's then finding the condition column, and it's isolating out any rows that have PPD present within that condition column. And this is going to be much more reliable, much more reproducible as far as not encountering errors throughout the process. And just like we saw with select, and like we saw with relocate, and like we saw with rename, we can actually filter things that are either or different particular values. And the way we would do this is we just need to modify it. Instead of being a single argument, it can now be two arguments. So kind of like we saw for week 1, we can use the little C function, which stands as concatenate, place the two values we're interested in together, and then it's basically going to be checking the condition column for anything that's either PPD or that's SEB, and it's going to return them both accordingly. So you can see in this case, this filtering ended up grabbing both PPD and SEB containing rows for the column condition. However, that can clutter things up, especially if you have multiple conditions that you're trying to filter for. So oftentimes I'd recommend just setting up that vector externally. So taking that same line of code that used to be within the parentheses, we can assign it to an object or a variable in our environment, and then just provide the object or variable in as part of this argument here, in which case filter is going to check the condition column for anything that's present within these conditions, referring back to your local environment, knowing that the values within these conditions are either PPB or SEB. Now, this is great if you only have a limited number of values that may be present in a column, but similar to the situation with the 199 columns, if you had like 199 different values that could be represented within a particular column as far as the value of the row, you don't want to have to list them all individually just to exclude one particular type. However, as we encountered during the conditional section for file paths, if you add a little exclamation point in front of a logical value, it will flip it. So in this case, if I created a variable which is called is this a spectral instrument, and I set the value to true, if I put the little exclamation sign in front of it, it's going to flip it and it's going to return it as false. So we can rely on the same mechanism within the dplyr package by just throwing in a question mark within the filter parentheses, and instead of selecting for a particular row that contains a value that we want, it will exclude that row with that particular value. So for example, if we didn't want to include the SEB condition within our final data that we're tidying up, we just throw that little exclamation sign right in front of the actual column name being searched, in which case, if condition is matching to SEB, and it finds a row that does match that condition, it's going to say don't actually keep this as far as flipping it with that little exclamation sign, and the SUB column is not actually going to be contained within your new data frame. So just like we saw before, if we pull all the resulting objects out of the condition column and we're checking with unique, you can see that we ended up filtering control rows as well as PPD rows, but we didn't end up including any of those SEB rows at that point. Similarly, we can also end up doing that same thing with the exclamation point to exclude columns that we don't actually want to include. So for example, if we're working with that bigger data frame object and we just didn't want to include that time point column, we can throw in the little exclamation point in front of the column name, and it's not going to be included within the actual subsetted down data set. All right, so at this point, we've learned a handful of functions from the dplyr package. We now have building blocks that allow us to rearrange and subset the larger data frame to whatever kind of format we want to actually be using it. But we still need to figure out, is there a way that we can create new additional columns or change or modify existing columns that we already have? And we'll need to be learning about how to do this with the mutate function. But before this, let's go ahead and slim down our current data frame, make it a little bit more easy to work with, have fewer columns, have fewer conditions, just so that we're able to visualize it all within a single view, not have to subset the number of rows or the number of columns. So this is once again an example of what a pipe situation would be, and we're going to take it starting just to the right of the assignment arrow. We're starting with our object data. We're making the contents of data available to the filter function through the pipe, at which point within the filter, we're looking for the condition column, and we're isolating out the rows that are matching the value of control within that condition column. So at that point, we'd have a data frame that still has the same number of columns, but has been subsetted down to only contain values that within the condition were equal to control. At that point, this intermediate object gets passed to the next filter function, at which point we're filtering down for the time point column, matching only those that have a value of 0, which corresponds to the cord blood only specimens. So once again, we are reducing further the number of rows that are present within our intermediate object. This is then passed off through the next pipe to our select function, which is acting on the number of columns. And we're saying we don't actually want the 19 or so columns. We only want to keep the bid, the time point, the condition, the date column, the T-cell count column, and the CD45 count column. So at that point, it's going to just select those columns, and it's going to be much narrower as far as the width. And then that's being passed to the rename function, which is just going to rename condition as lowercase condition, and then it's going to rename the bid column as the specimen column, just so we don't get confused. And then for the final passing through a pipe, it goes to relocate, which are basically saying, hey, please go ahead and place date after the specimen column. So if we ran this big chain of pipe commands passing the object to each other, we end up getting tidy data back that looks kind of like this, being assigned to our tidy data object. So as you can see, by combining these different pipes and these different functions together, we can do a lot of series of operations very, very quickly to get to the tidy kind of data that we're actually trying to work with. So looking at this particular column, we need to learn how to use the mutate column. And what we're going to be targeting is this T-cell count column and a CD45 count column. You can kind of see the T-cells are a smaller number of cells compared to the CD45 gate. So if we're trying to get back to proportion, what we want to ideally be able to do is divide the number of T-cell counts by the corresponding CD4 count column to get back the actual proportion of T-cells within that CD45 gate. So the way we'd be doing that with the mutate function is going to look something like this. Once again, the contents of tidy data are being made available to mutate through the pipe. And then at that point, we need to specify what we want actually done on the other side of the equal sign to the right, in which case we're saying the T-cell count column, please divide it by the CD45 count column, which is going to return to us a proportion. And then we're going to assign this to a new column, which we're going to call, in this case, T-cells underscore proportion CD45. So if we run that line of code, what we're actually going to see is now we have this new column appear, being called whatever we decided to call it, T-cells underscore proportion CD45 in this case. And then if you double check, you'd see all the math has been carried out, dividing the T-cell counts by the CD45 counts to give us back the proportion. However, if we look at those numbers, we have a lot more significant digits than we'd ever actually need to be used. So let's go ahead and try rounding that up so we have something that's much more easy to work with in our case. So the way we can do this is through the round function, which we still need to integrate within that mutate statement as far as the syntax goes, so that it's able to operate and create a new column. So in this case, what's basically happening is we're taking that last column we generated, the T cells underscore proportion, CD45, and we're using that as an argument within the round function, which then is looking at this column and basically rounding it to 2 significant digits across the board. And then all the output of this particular operation is being passed to the new column we're creating, in this case, the T cells rounded. So as you can see, it ends up rounding and creating that new column, and this is now made available. Now, if we wanted it, instead of being a proportion, if we wanted it to be showing up as a percentage, we could repeat the same step using the T cells rounded column, just times that by 100 and assign it as a T cells percentage or something like that as a new additional column. So you can see you can kind of take the intermediate columns, change them as you go just to make sure everything is behaving correctly, and then at the end you have the actual data that you actually want to work with. A useful thing, having already derived this proportion column for the T cells as part of the CD45 gate, we may want to rearrange our rows to show either descending or ascending as far as the actual proportion of T cells for a given tube in a given specimen. So this is where that arrange function will come in. And basically the way we'd be doing this is either if we wanted to just arrange it in ascending order, we just use the arrange function by itself. But if you wanted to do it in descending order, we're using arrange and descend in combination. So in this case, I prefer to see descending proportions of T cells. So basically, I'm taking that intermediate object we just created. It's being passed through the pipe to the arrange function. And then the T cell rounded column is being sorted based on descending values. And then it's being rearranged accordingly in terms of the number of rows that we encounter. So similarly, if we wanted to filter, let's say we only wanted to grab the top T cell rows for specific specimens that had more than 30% T cells as part of their CD45 gate. In this case, we're working with cord blood, so there's a bunch of NKs running around. You'd see in this case, we can modify our filter function. So instead of matching a particular character condition, which is what we did with percentage and percentage, we can just say, hey, look at the T cell rounded column and only keep things that have a value greater than 0.3, in which case it's only returning the rows that are greater than 0.3 in this case. Now, if I was like trying to screen for batch effects or trying to screen for outliers, I could just simply grab those specimen IDs. So I can then go open my computer, pull up the correct FCS files, and validate, is this weird or was it a situation that the sample got cryopreserved badly and everything but a T cell ended up dying off. And that's where I could do part of the filter function just to isolate those that match my particular criteria I'm interested in, and then just pull the specimen ID column so I actually have the infant IDs I need to go up and look for. And finally, having done all these different edits and cleaned up my data set, I may want to save that so I can work with it later, or I may want to save it so I can pass it off to a supervisor. So this is a situation where not only being able to read in your own CSV file, but also export it might be useful. So what we can do is basically first, we need to create a new file name and a new location that we want to save this file to. So in this case, my new file name is going to be my new dataset, and just remembering to add the file extension at the end. Kind of what we saw at the beginning, I'm creating a file path to the data folder, and then I'm basically providing the new name I want to save it as there. And then I'm saving this particular as a variable object we're calling storage location, which I can then make available to the write.csv function, which is available through base R. And I'm basically giving for the first argument the tidy data that I want to save to a new CSV, as well as its storage location. And then I'm just setting the row.names argument to false because we don't actually have row names in this particular object, so it's not actually needed. All right, so our takeaway. In this session, we have started to look at the main functions within the dplyr package, especially in the context of trying to keep tidy data. We've looked at how to select columns, how to filter for particular rows, how to create, modify existing values within each of these, And we're going to continue to build upon these and how to use pipes throughout the course, as well as add in additional Tidyverse functions that we haven't had time to cover today, as they're appropriate because they're able to do a lot of things with rectangular and square-shaped data. And as we saw, this will allow us to quickly and extensively modify our own datasets, which cuts out the necessity of doing this all by hand and having to go through multiple hours of clicking and pasting just to get to what we actually want. Once we have it set up, we're able to quickly modify it, quickly get back what we need, and save it once again to a new CSV file. In terms of the goals of this course, I try to make sure everybody's trying to be as reproducible and replicable as possible. So I urge you, if you're trying to edit a CSV file, don't do it by hand. It may seem easier just to go in and modify a particular one value in a particular one cell. But we don't have an actual track record of what has changed within those CSV files. So if you're trying to document every step that happened in your particular workflow, you're missing out on the data that way. But then also, creating your own R script that's just going to be able to quickly do the same cleanup steps is very quick and very efficient. So if you just keep your R script set up, you don't actually have to modify the original data. Likewise, if you do want to work with an intermediate object that you have created, if you save it as something else and don't overwrite the original data, you still have both versions and you can choose to use them. And you have them when you actually go and are submitting a report or submitting a paper. You have both the original data set as exported, as well as the one that you actually used for your analysis. So this is getting close to the end of the Intro to R focused lessons at the beginning of the Cytometry in R course. We still have one more as far as talking about how to use ggplot, but we're going to start pivoting more and more cytometry focused material over the next couple weeks. And this is especially the case next week, where we start looking at how to use gating sets and also looking at the main controversy as far as flow cytometry packages in bioconductor structure, as far as whether are they using flow frame objects or are they using cytoframe objects, which we'll go into as far as like what these actually are, what this actually mean, what the advantages of each is. So we'll be looking at that starting next week. However, I just want to preface that what we'll be doing for the second half is we'll bring in a workspace from FlowJo, in my case, into our actual R environment, just to show a case where it's more complicated gates, more complicated analysis, and how we're able to use this. Now, this is mediated through the CytoML package, which also is able to handle Diva and Cytobank kind of files. But if you don't have FlowJo, Diva, or Cytobank, and you have to bring in your own data set, I put out a pre-course walkthrough for Floriada.io, which will allow you to convert your own gating set for them into a FlowJo compatible workspace, so that you can actually use your own data set for this next upcoming week instead of having to rely on your own data set. So if you're interested in it, follow up with the walkthrough just to see how you'd be able to do that, and it's going to be a good week. And with that, we're at the top of the hour. I'm conscientious of everybody's time, so if you need to go, please feel free to leave. The additional resources for this week are two. So if you think back to my journey as far as learning R, after the disastrous initial first attempt in 2020, I found the Wellcome Trust's version of Intro to Bioinformatics, which was initial get me on the right path to learning. But then the particular aha moment that actually started clicking in the concepts of tidy data was through the Carpentries project, in terms of their data organization and spreadsheets and their data analysis and visualization in R, self-walkthrough courses that are available through their website. These are primarily aimed at ecologists, but we are handling data in very similar ways of trying to manipulate rows and columns, and these were particularly well done, so I'm recommending them to anybody who's interested in trying to work through these more additionally on their own. I've already mentioned the Rifamonas Project's YouTube channel. They have a lot of different videos as far as working with Tidyverse packages, and especially dplyr, so I linked their playlist for this particular one. Another YouTube channel, Simplistics, is more of a psych class statistics kind of event, but they do a very good job at their explanations as well, so I'm including them. This one was more in the old style of using select with the two arguments, but the next video afterwards shows how to use the pipes. The take-home problems this week are very much focused of I want everyone to be able to play around with their own dataset or play around with the dataset that we provided, just to get familiarity with how to use these different functions, because these are functions that we will be using basically every single day throughout the rest of the course. So the sooner you get some familiarity with like some of how to use them and apply them to your own data sets, the better off you'll be in the long run. So the very first question is just basically play around with the data, rearrange it however you see fit, and save it as a CSV file. The second one is the mutate function. That's the one that I find beginners struggle a little bit more as far as the syntax of the arguments, as far as how to modify an existing column. So basically, if you look at the data set we provided, there's a bunch of double columns that have a lot of significant digits. Basically, just round them up to a couple number of digits and just save them again on the existing data frame, just so you get familiar with the process of how to use mutate. And then the third one is the bid, time point, and condition columns originated originally from a file name argument within the CSV files. Basically, instead of having split them up, I want you to try to combine them back together using the paste function, and then save them as their own particular column, and then just double check that none of the new IDs you generated within that column are going to conflict or they're not unique with each other. And with that, that is all I have for you today. If you have any questions, I'll be in the chat for a little bit. If not, please reach out on the discussion forum. If you did submit take-home problems for the last couple weeks, I did not forget about you. You're on my to-do list for the remainder of the week. And I will also link again to the how to submit this homework for this week so that we don't run into merge request issues when you try to pull in data for next week. And I wish everyone a good day, and thanks for paying attention. And Carlos, I also still learning new Tidyverse dplyr functions to this very day. I every time I open up like the little help side of dplyr, there's like another 5 functions. I'm just like, what does this do? And then I'm like, why didn't I know this like a year and a half ago?